All right. Well, thank you everyone for joining us today, especially with all of the bad weather on the East Coast and the Mid-Atlantic and Southeast and pretty much everywhere. So thank you for taking the time to join us today. My name is Jana Foster. I am the engagement manager for MARACOOS. And we are going to have Tina Sepapor do an introduction for MARACOOS. Um, so if you go ahead and take it away, Tina, thank you so much.
Awesome. Thanks, Shanna, and thank you everyone for attending today. I will be giving you a quick overview of MARACOOS, which is the Mid-Atlantic Regional Association Coastal Ocean Observing System, before we dive into our speaker for today. So, MARACOOS is a nonprofit organization, excuse me, and is one of 11 regional associations that make up the U.S. Integrated Ocean Observing System, also known as IOOS. We serve the Mid-Atlantic region, covering 10 states from Cape Cod, Massachusetts to Cape Hatteras, North Carolina. This region includes 5 major estuaries like the Chesapeake Bay and supports a population of over 78 million people. The Mid-Atlantic is rich in ocean and coastal data, and MARACOOS makes that data available to be turned into usable information that supports safe navigation, emergency response, environmental health, resource management, and economic planning. We collect and serve our own near real-time observing data while also integrating data from state and local agencies, universities, and other partners. These community and institutional collaborations are essential to understanding and responding to the unique needs of the Mid-Atlantic. All MARACOOS data are federally certified, meeting rigorous standards that ensure the quality and reliability required for high-level decision-making at local, state, and national levels. Our set of tools, including our Ocean Map data portal, is robust, user-friendly, and highly customizable. If you are interested, please feel free to reach out. We'd love to connect, learn more about your needs, and walk you through how these tools can be customized for your region. We are a stakeholder-driven organization built on a network of community partners and local experts. This allows us to stay closely connected to the regional and local priorities of the Mid-Atlantic, ensuring our efforts are responsive, relative, and grounded in real-world needs. Rather than duplicating these efforts, we focus on amplifying the work already happening across the region. Our goal is to deliver the highest quality ocean and coastal data to support effective, informed decision making. We organize our work around 5 focus areas that reflect the full range of needs across the Mid-Atlantic. These focus areas include coastal hazards, maritime safety and commerce, fisheries and natural resources, water quality, and energy. If you have data to share and are interested in partnering with us, we'd love to hear from you. You can reach us anytime at info@maracoos.org. And with that, today's webinar will be highlighting how eDNA is being applied in real-world research and monitoring, and how these tools are helping scientists better understand and track changes in aquatic ecosystems. So now for our first speaker is Dr. Matt Ogburn, a marine ecologist and senior scientist leading the Fisheries Conservation Lab at the Smithsonian Environmental Research Center. His lab focuses on understanding the biology and ecology of fisheries species, the ecosystem that support them, and how this information can be applied in management and conservation. He applies environmental DNA approaches to a range of topics, including evaluating responses of migratory fish to dam removal projects, food webs, and biodiversity monitoring. He also leads the Chesapeake Bay Barcode Initiative, which is compiling barcode libraries for fish and maritime macroinvertebrates for the Chesapeake region.
Great. Thanks so much, Tina, and thanks everyone for joining today. Um, Hopeful that we didn't lose too many people to the weather challenges, but I know it's a tough day out there. Um, yeah, so I really appreciate this opportunity through Maracoo's to share some of the really applied work that we've been doing at Smithsonian, developing and applying environmental DNA tools to various topics in fisheries ecology. I wanted to start if I can get my slides to advance. I wanted to start with just a brief overview of environmental DNA. I imagine a lot of you are familiar with it, but just so that we're all starting from the same place here. Um, Environmental DNA is generally refers to approaches that allow us to monitor and detect the presence of species based on molecular detection of aqueous DNA. So we're using genetics, molecular tools to detect DNA in the water. People also apply it in terrestrial systems to things like gut contents of fish, which will be one of my examples, and even collecting DNA from the air, from aerosols as well. So there's a lot of different sources of DNA that you can use these approaches for. But today we're especially talking about DNA that's in the water. Um, Environmental DNA is typically either nuclear or mitochondrial DNA that is released from a plant or an animal into the environment. It might be from feces, from mucus, from gametes during spawning, from skin or hair that are shed, and even from carcasses. So there can be a lot of different sources of DNA into the environment. In aquatic environments, that eDNA, not surprisingly, is diluted by the water. And it also doesn't persist forever. Generally, in the water, it can last something like 7 to 21 days, depending on the conditions. But that's affected by things like heat, um, the pH of the water. Lots of different things can affect what that what that duration of eDNA is in the environment. Um, but nevertheless, it's the signal that's out there that can be detected using genetic approaches and gives us a lot of opportunities to learn about what's going on in the environment, who's there, what their relative abundances are, and a lot of other things. I've been looking for good ways to sort of relate it to something, what's a good metaphor for environmental DNA. And one of the ones I saw recently is someone walking their dog around their neighborhood, where their dog doesn't necessarily, the dog might get excited if it sees one of the other dogs in the neighborhood or one of the other people, but it also gets excited if it smells them, even in really low concentrations. And so you can sort of think of that in a similar way as eDNA, where the animal that produced it or might be there, or it might have been there recently, and all of that is useful information for understanding the environment. There are two different approaches in particular that I'll highlight in my talk today. That are both environmental DNA approaches, but coming at it from sort of a different direction. One is the development of targeted genetic probes that target individual species or groups of species, where you might be looking for something that's rare in the environment and you want a highly sensitive tool to detect that rare thing. Or meta barcoding, where you're basically looking at the genetics or the genetic barcode for everything in a sample, or large groups of things like fish or invertebrates in a sample, and looking at the composition of that community of species. So those are sort of two different approaches, different genetic tools that each are really valuable in environmental studies. In the introduction, Tina mentioned the Chesapeake Bay Barcode Initiative, and this is an initiative that I've been co-leading with colleagues at the Smithsonian to really develop the sort of the library of species genetic information that can underpin environmental DNA studies in the Chesapeake and Mid-Atlantic regions. So when you use these genetic tools in environmental DNA approaches, you get back lots of sequences of A's and G's and T's and C's um, that you can differentiate and group, and that might tell you that there are, hundreds of different types of sequences in your sample, and some are common and some are rare. Uh, but without a library of genetic sequences or barcodes, you don't know what those things are. And so we've been developing that library on the back end that really supports rigorous environmental DNA studies in this region. And one of the ways that people do that is to go back into historical museum collections. And so if you think of like all of the birds or all of the mammals that have been collected at Smithsonian over the last 200 years, um, you know, there's lots of different skins of animals that you can go back and sample, and that works great for some things. But most marine animals, fish and marine invertebrates, were preserved using formalin or formaldehyde, which greatly degrades their DNA. And so we can't really go back to those samples and use them reliably. So what we've been doing for the last 12 years or so is working with a lot of different groups in the Chesapeake region that go out and regularly sample marine animals and have been recollecting 3 to 5 individual specimens of all of the species in the region. It's a little bit over 300 fish species in the Chesapeake tidal waters, from salty to freshwater tidal. And we've collected about 90% of those species. And for invertebrates, it's somewhere in the ballpark of 1,000 or so species, and we've collected over 500 of them, including the vast majority of common things like clams and crabs and shrimp and things like that. So we've been making really good progress in building up this database that's getting close to 1,000 species that we've collected, and for which we can find out detailed information from environmental DNA approaches. Just to give you a sense of what that has looked like, um, for each of the specimens that we collect, we take a photograph, we take a genetic sample that we use to get the DNA barcode. Um, One of the things about DNA studies is that you have to pick regions of the DNA of an animal to study. And for eDNA projects, typically you look for a region that is variable enough that it varies among the different species that you're interested in, so you can tell them apart, but not so much that individuals within a species are different. Um, so we use, for our barcode library, we've used 2 different sequences. One is the CO1 sequence, which gives you a broad sense of both fish and invertebrates. And then for the fish samples, we've also used part of the 12S um, sequence, and that's a more targeted sequence for fish. There are other ones out there for for invertebrates and fish and other animals, but those are the ones we've used for our data set so far. Um, we collect a frozen tissue sample as well that goes into Smithsonian's frozen tissue biorepository if people want to go back and study that tissue with new sequences or using new techniques. And then, assuming it's not so small that we used the whole thing to do those things, we save a physical voucher specimen that's in the National Museum of Natural History as well. So this picture here shows from one of our collection efforts a pipeline of people starting with sorting and identifying in the foreground, working into the background towards collecting those tissue samples and DNA samples to sequence. Those are all publicly available in the in NCBI bioprojects, which is a database where you can go find out the sequence information, and we have separate projects for the fish and invertebrates. And I'm happy to talk with you, or Rob Aguilar is as well, who's in my group, if you want to know more. So the 3 examples that I want to give today um, are ones using really targeted genetic probes. Um, those focus on conservation of river herring, which were in my title slide photo. We've also used metabarcoding approaches for studying the stomach contents of striped bass. And third, I'll get into a new project we have going on with MARACOOS and a variety of partners called the Mid-Atlantic Marine Biodiversity Observation Network, in which we're using meta-barcoding of water samples to study biodiversity in the Mid-Atlantic region out on the continental shelf. To dive into the river herring conservation section of this, we developed this set of work for a range of reasons, but one in particular is that these two species of river herring that we have, alewife and blueback herring, which I'll have pictures of again in a minute. They're anadromous fish, which means they live mostly in the ocean and migrate into freshwater to spawn. And their populations have had something like a 90% decline over the last half century or so, due to overfishing and various other causes, including the construction of dams that blocked them from being able to get to their former spawning grounds. And one of the less publicized but really important parts of the Chesapeake Bay restoration program has been focused on the removal of dams that are no longer in use, to open up habitat to anadromous and other migratory fish, to habitats that had been blocked off sometimes for over a century. And so we started studying river herring in part to help document whether they, how they responded to these dam removals, whether they were able to move into habitat that was open back up. And one of the examples that we have is this dam pictured on the left, Bloody Dam. It was built in 1907 in Maryland. It's in Patapsco Valley State Park, and really close to Baltimore Washington International Airport, just to give you a sense of where we're talking about. Um, it it operated as one of an early hydropower dam for about 30 years before the pond behind it was completely filled in. And it doesn't look very big in this picture, but it was about 3 stories tall. So it's a pretty large dam, and was the first dam on the Patapsco River the most downstream dam that blocked fish migrations. Um, So the dam removal was a large project by a lot of the different partners, whose logos are shown here at the top right of the slide. And when that dam was removed in 2018, it restored about 65 kilometers of stream access for migratory fish and other migratory organisms. So the picture on the right is from just about the same perspective as the picture on the left, showing that there's still some little rapids there, but the dam and the obstruction is gone, allowing fish to move past that area if they choose to. So for this project, our key question really was assessing whether removal of this dam restored river herring spawning habitat use upstream of the dam. And we did that in 2 ways, and actually a couple others. We wanted to integrate across multiple methods to really do the best we could at answering this question. And different approaches have different strengths and weaknesses. And so for what I'm showing today, we combined environmental DNA or eDNA approaches with a more traditional approach where we put a plankton net in the stream and collected ichthyoplankton, basically eggs and larvae of river herring, and counted those things. The eDNA let us know whether river herring were detected above the dam site after removal. It's more of a presence absence, and maybe some sense of relative abundance. And it's species specific. So we were able to tell if it's an alewife or a blueback herring or something else, but focused on those two species. And then the ichthyoplankton were nice to add on to that because it would tell us if there's actively spawning going on, if there's eggs or not. And but one of the drawbacks of that is that it's not species specific. So they could be river herring eggs, but those also look really similar to hickory shad eggs and gizzard shad eggs. So we aren't able to, we can tell there's spawning going on there, but we don't know for certain the species unless we do the genetics of every individual egg, which gets expensive really quickly. So here's a map of the Patapsco River with Baltimore, the city and port of Baltimore on the right-hand side. And then if you were a migratory fish, you would be coming from that port area and going from right to left across the map. Um, we divided the river into three different sections for this study. The kind of pinkish color, the downstream circles here are sites that were downstream of Bloody Dam. And we monitored those sites to see, we wanted to know if river herring were present anywhere in the river. We had done some preliminary sampling and knew that they were there, but that sort of documents what's happening below the dam. Then we had this stretch with the orange triangles that we called the restored sites that were between Bloody Dam and the next dam on the river, Daniels Dam. And Daniels Dam did have a fish ladder on it. Um, And so there is the possibility that if fish made it there, they could continue further upstream to the green squares sites above Daniels Dam. So we have these three different sections of the river that we were monitoring both before and after the restoration. What the sampling basically looked like was taking a 1 liter bottle, um, putting a glove on your hand, carefully wading into the stream so that you didn't stir up tons of sediment and stuff, and holding the bottle under the surface upstream of you so you didn't contaminate the sample, and then capping it off. And so that was how we collected the samples. So it's a pretty quick, simple thing to do. We did that depending on the year, either every week or every 2 weeks. Um, and also, depending on the year, we either took single samples or duplicate samples, depending on how much funding we had available. Um, but and the spawning run for these fish, the alewife come in first. They show up generally in February or early March, and then the blueback herring show up a few weeks later, and the run of both species typically is done by June 1st. So we sampled from kind of late February, early March through May for this study. And this is what the data looked like from 2015 to 2021, which was 4 years of sampling prior to the dam removal and 3 years of sampling after the dam removal. And this is for the egg counts from our ichthyoplankton samples, where we put a plankton net in the water in the stream for 5 minutes at a time, and then sorted through that to count the number of eggs present. So on the panel on the left, on each of the figures that I'll show is the before times, before the dam removal, and the one on the right is after the dam removal. There are some sites that look like just white circles. That's where eggs, or in the future slides, DNA was absent. The ones that are colored in are presence of eggs or DNA in the next slides. And the size of the circle that's colored in gives you a sense of the relative abundance of eggs or gene sequences that we get. Some of the dots have X's across them as well before the restoration, and those were sites that were not sampled during that time period. So we didn't sample all of the sites every year. But basically what we saw for eggs is that both before and after the dam removal, at least through 2021, we didn't see, we collected eggs downstream, sometimes thousands of eggs per 100 cubic meters of water, but did not detect any eggs in the restored reach either before or after the restoration. But as I mentioned before, some of these targeted eDNA probes can really be really sensitive. And so we thought, detecting presence of fish might be more possible than catching eggs. And that turned out to be the case. The genetic probes that we used were developed in collaboration with Lewis Plow, who used to be at the University of Maryland Center for Environmental Science Horn Point Laboratory. He's since moved on to other things. But this is one of the projects that we deployed those probes on. So again, if you look before the sampling in the left-hand panel, you see some yellowish dots for alewife that are only downstream of Bloody Dam, but not any of the sites upstream. And then after the dam removal, which actually started the very first year, 2019, after the dam removal, we didn't see large concentrations of DNA upstream of the dam in that restored reach between the two dams, but we did see some DNA there. We did not see any evidence that alewife were using the fish ladder at Daniels Dam and moving further upstream, but they were present in that restored reach. Um, the story was a little bit different for blueback herring. The numbers of blueback herring in the system are larger in general, and we know that because we've been doing run counts as well using other methods. So the concentrations of eDNA for blueback herring, both before and after the restoration, were higher. And after the restoration, blueback herring were found both in the restored reach between the dams and then also upstream of Daniels Dam, showing that they were able to use that fish ladder and move up into the upper parts of the river, which was really exciting. So we're you know, the evidence from the eggs and particularly from the DNA showed that starting the very first year after the dam removal, we were able to detect fish moving upstream. We did, I don't have a figure that looks quite the same here. We did continue this sampling for 2 more years, and we did detect eggs in the restored reach in 2022 and 2023. So we do now have evidence of spawning in that restored reach, just not in the figures from this paper that I have cited here. But we've found that combining this environmental DNA approach that's really sensitive gave us a really early first look at how fish were using the system following dam removal. And then adding in the egg sampling allowed us to also confirm that they're using that restored habitat for reproduction. Um, so we've used these probes for a variety of other studies too, and I just wanted to highlight one briefly. One of the big questions with eDNA sampling is you sometimes get really low concentrations and sometimes really high concentrations. And we really want to know if that equates to numbers of fish or if it's just sort of by luck or, you know, something else. So we have done a study also working with Lewis Plow's group and a graduate student, Chelsea Fowler, um, to really test that question. We did this in the Choptank River, which is also in the Chesapeake Bay watershed, where we used this automated water sampler in the picture on the top left to collect duplicate water samples every day. Sorry, triplicate water samples every day. And then we also used a high resolution Didson imaging sonar to record and count the fish passing the same location each day. Um, and Chelsea did some really great work comparing the counts of fish to the concentrations of DNA in the water, and also accounting for things like flow rate and temperature and DNA degradation rates, and showed that she had a model that was able to use that information to model the population there, the run count, and able to estimate it within 0.5% of the total run count. So this study and others that other people have published are showing that we do think we can get at relative abundance with eDNA in systems like this as well. It's got to be sort of the right conditions and right system, but we think that it can work pretty well. So moving on to the second sort of approach here, the meta barcoding approach, looking at the community. This is a study we conducted with Dr. Katrina Lohan, who's one of my colleagues at Smithsonian. And the two goals of this project were really to identify key prey items for juvenile striped bass. And Maya in this picture is holding a larger juvenile, but these went all the way down to juveniles that are just an inch or two long. Fish that are sampled in the young of year surveys that are used to predict the future population size in the Chesapeake, and for which there was very limited diet data across the Chesapeake tributaries. Um, because my colleague Katrina is also interested in the role of parasites in food webs. She leads our coastal disease ecology lab. We were interested in seeing if in doing this study for prey items of striped bass, we could also detect parasites using genetic methods. So for this study, we obtained the fish primarily from the Maryland and Virginia seine surveys that target juvenile striped bass for population studies. And we supplemented that a little bit by adding a couple other sites and then adding some gillnet sampling for larger juveniles than what they typically collect in the seine surveys. So we ended up with young of year fish, the really small ones, from 9 different tributaries. We had roughly age 1 to 4 fish that we collected from gillnets in 2 tributaries on the eastern, one on the eastern and one on the western shore of Maryland. Um, and then we also were able to obtain some adults that were from the adult spawning stock survey that Maryland Department of Natural Resources conducts that they provided to us. So for each of these fish, we dissected out the stomachs. They literally went in a blender. And then from that slurry, we extracted DNA. And this, I'm not going into super detail on methods and results, but wanted to show you the kind of information that we're getting from this. This study was published in 2023 as well, if you want to learn more. But what this shows is the results, a summary of the results from the prey item question here. The figure on the left with the 3 columns shows the stomach content results from DNA. This is using the CO1 sequence that I mentioned before that we have in our Chesapeake Bay Barcode Initiative. And it's broken down, each bar is a different salinity range. So where it says sample down at the bottom, those are the salinities that the samples were from. So the bar all the way on the left is essentially freshwater, and then slightly brackish water, and then sort of moderate salinities. And we see different distributions of prey across those. And I wanted to highlight some of the most common ones. In the freshwater zone, the most common types of sequences that we got back from this are from dipterans, basically insects like flies and other sorts of related insects. And so that's what the picture that's labeled 1 is an example of the aquatic phase larvae of a fly that these fish are not jumping out of the water necessarily and catching the adults, but a lot of dipterans have these aquatic larval stages that the fish are eating. As you move into the slightly salty kind of middle bar, one of the most common ones are the mysids, which are shrimp-like invertebrates. Photo number 2, really common prey item there. And then as you move into some of the saltier parts of the bay, the most common sequences that we got were from amphipods, photo number 3, which are also different small invertebrates that make up a lot of the prey items in there. And so we're seeing a lot of different things. If you sort of use your compare the colors, you can see some of the other things that were relatively common. But showing you that this meta-barcoding approach is giving us really detailed information. And I'm not showing you all the detail here. For most of these, we're getting species level information. So when it says fish other or invertebrate other or bivalve other, usually we know the exact species name of the things that are being eaten. And even picks up some things that you might not otherwise think as prey of a 2-inch long striped bass. Horseshoe crab comes up pretty commonly, and barnacles also. Um, and in both of those cases, the fourth photo that doesn't have a number here is the larval stage of a horseshoe crab. So, we did pick through some of the stomachs just to uh, under a microscope to try to get some information to confirm what we were seeing in the genetics. And we see both, larval, a lot of larval horseshoe crabs, and also a whole bunch of larval barnacles as well, which are planktonic. So things that you would expect a fish that size to eat. So the genetic data is proving to be really powerful and helping to get this diet data for small fish, which otherwise is really challenging and difficult and time consuming to do um, under a microscope, and gives you much more detailed species level information on what they're eating. Um, on the parasite part, for the parasites, we needed sort of a broader um, community look. So we used a different genetic probe that targets the 18S gene, and that gives us a wide range, a lot of the same things, but also a wide range of parasites that infect striped bass. And I've just got pictures of a number of them here. In this figure on the left, the x-axis is different rivers that we sampled, and then the y-axis is relative abundance of those sequences from each river, and then the three panels go from freshwater at the top towards the saltier areas at the bottom. And so you can sort of see that there's a mix of different things, but a whole lot of uh, a whole lot of arthropod and other kinds of things in here. But I'll just highlight that some of the parasites that we detected are ones that affect striped bass, but some of them are ones that don't infect striped bass, but do infect some of the prey items that we detected, um, which suggests that those parasites might play a role in in potentially even making prey more vulnerable to capture by striped bass, because there are a lot of parasites that are known to modify prey behavior in such a way that they are more likely to get eaten, so that that parasite can move through the food web to complete its life cycle. So we see some of those showing up that aren't parasites of striped bass. So there's another follow-up paper that digs into the parasite part of this in more detail that we're working on right now. But some really interesting results on the parasite side as well. So finally, getting into the other metabarcoding example here, the Mid-Atlantic Marine Biodiversity Observation Network. What we're using for this is basically the same approach as the striped bass stomach contents, except this time with water samples. And the person sitting in the middle here is Peter Thielen, who I don't know if he's had the chance to join us or not. I know he was having some weather issues this morning. Um, but we're part of a group that's led by MARACOOS that is sampling a line of sites. And I have a map on the next slide that extends off of Ocean City, Maryland, going about 2/3 of the way across the continental shelf. And um, I'll just point out that this is our most inshore sampling station. So you can see Ocean City in the background. Um, but the goals of this project, which is a 4-year project, we just finished up the first year, um, are to integrate information from the environmental DNA samples, from animal telemetry, where we're tracking individual animals using acoustic telemetry tags or satellite telemetry tags, but able to track individual animals, and also passive acoustic monitoring, where we have hydrophones or underwater microphones that are listening both for all sounds that are in the environment, and also ones that particularly are listening for cetaceans, whales, and dolphins, um, to be able to collect information on them. And then we'll be developing modeling products that integrate those different types of biodiversity monitoring information, and turn those into data products that can be used for management. Many of the species in this region are ones that are highly migratory. So one of the planned outputs of this project is a series of maps at monthly timescales that integrate oceanographic data and the biodiversity data and make detailed predictions about where key species ranging from things like sturgeon to sharks to whales um, are occurring in the mid-Atlantic and particularly in this region off the coast of Maryland, when they're likely to be there, how close to shore or far off of shore, and how quickly they're sort of passing through this region. Um, there's a lot of use of this region by different stakeholders, whether it's planned offshore wind developments, which are the series of gray dots in the middle of the picture, whether they are maritime trade, the boxes towards the top, the dashed box shows trade routes of ships coming out of Delaware Bay. There's a lot of different activities, a lot of commercial and recreational fishing all going on in this area. And so we're hoping to be able to integrate these different kinds of data that include environmental DNA data to really develop products that can be used to help manage for those various uses of the region. Again, thank you for the opportunity to share this work. We've had funding for the projects that I've been talking about from a number of different sources. The Smithsonian Institution Barcode Network and Global Genome Initiative have been supporting the Chesapeake Bay Barcode Initiative. National Fish and Wildlife Foundation supported the river herring work, along with NOAA Chesapeake Bay Office. And the National Ocean Partnership Program and NOAA's Marine Life Program have funded this Mid-Atlantic MBON project as one of 10 MBON projects in different regions around the US. So if I can't see who's on here at the moment, I'm going to stop sharing real quick. If Peter's joined, we'll pass it off to him. And if not, we can wrap up there and I'll take questions.
It seems like Peter was unable to join today with all the bad weather, which is so understandable. And we
Mhm.
appreciate you being here today, Matt, and all of you joining us. But we'll go ahead and do some questions. I have a couple
Mhm.
that came in advance, so I'll ask those. And if anyone else has questions, please feel free to either raise your hand or drop them in the question and answer function on Zoom. One of the first questions I got, and I know that with the dam, that was a really good like long-term example of evaluating and monitoring an ecosystem before and after. But is this possible to use this technology, these insights to monitor before after like a short-term disaster, like a hurricane or an oil spill or something along those lines?
I mean, yes, potentially. I think for environmental DNA approaches, the question has to align with the technology. And so, what one of the things that's made this work well for these anadromous fish is that they're only present in the river for a few months of the year. And we've done some sampling throughout the rest of the year, and we don't detect their DNA, coming from the sediment or other things. So um, it's from that perspective, what we see is kind of a clean slate every year, and so we're able to detect these changes from year to year in where fish are using the habitat that particular year. Um, if you were looking for, striped bass in Chesapeake Bay and just looking for their presence, they're probably going to be there before or after a major hurricane or something. So it would need to be, there would need to be some signal that you would expect to be different in the DNA. And so, it might be a change in community composition where certain species disappear or become much less abundant. You might be able to pick that up. But you need to put some thought into what the signal is you think you might see and develop a study around that design. Yeah.
Wonderful. And then we've got a question here from one of the attendants. We're gonna allow them to ask that question. There you go. Hi, Menon. Okay. Well, I'll go ahead and get to the next question and we'll see if we get that question. Um, let's see in the chat here. What's the possibility of using nuclear eDNA to do more in-depth analysis? mtDNA is more abundant. However, I believe technology should be adaptable to nuclear DNA.
Yeah, no, that's a great question. And I wish Peter was here to answer that one. But some of the approaches that I've been using in these particular studies are mitochondrial DNA. Um, there are a lot of sort of, DNA technology is improving really rapidly. And so, some of the the technology that's available that's being used in eDNA studies that are starting within the last few years can leverage approaches that do look a lot more, look at nuclear DNA and look at across the whole genome rather than picking out these targeted regions of mitochondrial DNA. And so that's something that we've been talking a lot about, like for this Mid-Atlantic Imbond project is something we might explore. Um, but the technologies are emerging now to be able to really look across the whole genome and using nuclear DNA that can give a lot more detailed information. Um, and that's one of the reasons, going back to the first part of my talk, one of the reasons why we save these tissue samples in a biorepository, um, so that we can go back to those and, as people start to apply, new approaches to environmental DNA, we can go back to those same samples and just add that information to the barcode library as well, so that um, they're comparable.
Wonderful. Thank you, Matt. And then we got another question here in the chat. Are CBBI or SERC or MBON protocols publicly available for scientists to do similar work? Um,
Um, so yes. So the Chesapeake Bay Barcode Initiative, sort of approaches that we used, we're happy to share that. The data are all publicly available. Um, we're slowly adding to the database over time. So the most definitive copy can be obtained by emailing us directly. And we're happy to share like the latest version, but we a couple times a year update what's available online. But we're happy to share the actual, those are the data. We're happy to share the actual methods. Um, for the Inbond project, we're in the process of making our final decisions about what genetic markers we're going to use and exactly what platforms to sequence those on. And so that's you know, uh, something that we will share once we make that decision, but I can't tell you because we don't quite have the final decision on it. Um, but I think those they will be approaches that are, publicly available through, publications and, and even commercial sequencing operations. So those are, we're happy to share more information about that.
Wonderful. Thank you,
Yeah.
Matt. And Ava, if you need to, you can email the email address and I can try to connect you with Matt offline
Mhm.
after this. Um, how do you decide? Another question. How do you decide a species is cryptic or rare when there is still a lack of information in the library? What are the standard sampling volume in terms of soil, sediment, or water?
Oof, yeah. Lots of great questions there. Um, so yeah, I mean, I think it depends on your approach. Um, so if a species is rare and it's not been sequenced before, sort of the first step is to get sequences for it and add them to the database, so that if you detect that in some of your approaches, um, metabarcoding or otherwise, you know what it is. Um, so that's kind of a key step. Um, we do, we've been trying to do a lot of that. It takes a lot of effort with taxonomists when they're, but we do see evidence of, even in the Chesapeake region that's well studied, of of weird things that I'll just say weird things that show up in the samples. So, like there's a species of freshwater fish that we pick up that's often used as bait for recreational fishermen. And so we got in some of our collections, it looked like two different species potentially with the DNA samples. But what was actually happening was some of the fish were from like Chesapeake stock, and some were moved from the Ohio River, sort of the Mississippi River drainage. So they're the same species, but somewhat different genetically, and we pick things like that up. Um, and then sometimes there are potentially new species that haven't been described anywhere. So we've collected some sea cucumbers from out in the like right along the coast, and it looks like it's a suite of multiple species that also look pretty similar. So we think there's some there that haven't been described before. Um, but there's all kinds of challenges like that that pop up, and it takes a lot of time and detective work to figure that out sometimes. And there's even like there's one non-native shrimp that's been introduced to the region that we've been studying in detail, and it's this whole like laundry list of reasons that it's been really hard to study it because there are it's the sequences we've gotten matched to several different potential species that have been described in the past. Um, and they all look very similar. They're a little peppermint shrimp, so like a cute aquarium peppermint shrimp that's got red stripes on it. Um, and um, so we've tried to go back to those original museum specimens that were described from different places around the world. And one of them we couldn't go back and look at because it was lost in the Great Fire in Chicago. Um, One of them from Germany we couldn't see because it was lost in a museum during when Dresden was firebombed during World War II. So it's been this like whole saga of trying to go back and find the originally collected and described specimens of certain species that we've seen. Um, so that's been one of the most extreme examples. But yeah, so that's challenging. So I've only been sort of answering the first part of the question. As far as standard sampling volumes, there and things there's a lot of different approaches. So I would just say that it depends on what you want, and I would recommend looking at the scientific literature, and talking to people doing it to try to get the best sense of what might work for your particular study. Um, the sample volume is critical. The size filter that you filter that water through is important. The way that you extract the DNA from that sample that's been filtered is important. Um, so there's lots of different steps along the way. But there's several publications out there currently that sort of help walk you through that decision, and various other resources online. But it just, talking to people in detail about your specific project is a really good approach as well.
Wonderful. Well, let's get one or two more questions in that we've got that came through the chat before we wrap up. We've got one from Caitlin. You mentioned that for the bivalves detected that you didn't get any species hit. Do you think that's because of lack of available, or do you think it's because these are novel species?
I didn't, yeah. If I said that, I misspoke. The graph said bivalves unidentified or bivalves other is what it said. And that we would just grouped those together. So we did have species information for most of the bivalves. What was interesting and sort of surprising to us in that striped bass study is that um, it was the insects actually are the ones that we couldn't get species information on. We haven't been focused on them because they weren't in our group of marine invertebrates that we've been targeting through the Chesapeake Bay Barcode Initiative. And for whatever reason, people who study dipterans, like flies and their friends, um, haven't sequenced them with the CO1 barcode, at least at the time that we were doing that. Hadn't sequenced that region and made it publicly available. So we have a sense for what they probably are based on what the larvae looked like that we saw. Um, but in the genetics part, like the most of the sequences that we couldn't identify to the species level were insects, which weren't part of our barcoding thing.
Awesome. Thank you, Matt. And then it looks like we just got one more question from Tim. Did you use blocking primers to allow detection of other fish species when me metabarcoding these striped bass stomach contents?
Yeah, no, that's a great question. Um, I don't believe that we did in this particular study, although I would actually have to go back because I am not the genetics lab that runs these things. Um, we spent a lot of time talking about whether to do that or not, and so just so that others have a sense of what we're talking about, a blocking primer is one where in our stomach content project for striped bass, you would expect that you would get a lot of striped bass sequences because they're this we're taking stuff from the stomach of a striped bass, and so you can design primers that say, when you run the genetics that say, if it's striped bass DNA, ignore it, basically. Just do the other stuff. So we decided not to take that approach. Um, we obviously did get a fair number of striped bass sequences, but we tried to create, to have enough of what's called sequencing depth that we got, striped bass and lots of other things, and just eliminated the striped bass. So it's possible a striped bass could have eaten another striped bass, and we wouldn't really know that. You wouldn't know that either if you used blocking primers, but um, but we got enough sequences to be able to say a lot about the prey, in addition to getting some that were striped bass.
Wonderful. Well, thank you. And thank you, everyone, for your questions. I just want to say, first off, thank you, Matt, for being here. And thank you for filling the whole hour for us with Peter not being able to attend. So we really appreciate that.
Yeah.
And then thank you everyone for tuning in today. We really appreciate it. We know that with weather, it might not have been easier for everyone, so thank you for attending. This will be recorded and put on our YouTube channel. And then I'm going to share all of MARACOOS's social media links. If you would like to stay connected with us and future webinars, this is how you would do it. So thank you again, everyone, and have a lovely rest of your day. Thank you again, Matt.
Yeah, great. Thank you.